Epigenetics is the study, in the field of genetics, of cellular and physiological phenotypic trait variations that are caused by external or environmental factors that switch genes on and off in affect how cells read genes instead of being caused by changes in the DNA sequence. Hence, epigenetic research seeks to describe dynamic alterations in the transcriptional potential of a cell. These alterations may or may not be heritable, although the use of the term epigenetic to describe processes that are not heritable is controversial. Unlike genetics based on changes to the DNA sequence, the changes in gene expression or cellular phenotype of epigenetics have other causes. Thus, use of the prefix EPI. The term also refers to the changes themselves, functionally relevant changes to the genome that do not involve a change in the nucleotide sequence. Examples of mechanisms that produce such changes are DNA methylation and histone modification, each of which alters how genes are expressed without altering the underlying DNA sequence. Gene expression can be controlled through the action of repressor proteins that attach to silencer regions of the DNA. These epigenetic changes may last through cell divisions for the duration of the cell's life, and may also last for multiple generations even though they do not involve changes in the underlying DNA sequence of the organism. Instead, non-genetic factors cause the organism's genes to behave differently. One example of an epigenetic change in eukaryotic biology is the process of cellular differentiation. In other words, as a single fertilized egg cell, the zygote, continues to divide, the resulting daughter cells change into all the different cell types in an organism, including neurons, muscle cells, epithelium, endothelium of blood vessels, etc., by activating some genes while inhibiting the expression of others. Definitions. The term epigenetics in its contemporary usage emerged in the 1990s, but for some years has been used in somewhat variable meanings. A consensus definition of the concept of epigenetic trait as stably heritable phenotype resulting from changes in a chromosome without alterations in the DNA sequence was formulated at a Cold Spring Harbor meeting in 2008. Historical. The term epigenesis has a generic meaning, extra growth, taken directly from Koine Greek pi iota gamma epsilon nu eta sigma iota sigma, used in English since the 17th century. From this, and the associated adjective epigenetic, the term epigenetics was coined by C. H. Waddington in 1942 as pertaining to epigenesis in parallel to Valentin Hecker's phonogenetics. Epigenesis in the context of biology refers to the differentiation of cells from their initial totipotent state in embryonic development. When Waddington coined the term, the physical nature of genes and their role in heredity was not known. He used it as a conceptual model of how genes might interact with their surroundings to produce a phenotype. He used the phrase epigenetic landscape as a metaphor for biological development. Waddington held that cell fates were established in development much like a marble rolls down to the point of lowest local elevation. Waddington suggested visualizing increasing irreversibility of cell type differentiation as ridges rising between the valleys where the marbles are traveling. In recent times, Waddington's notion of the epigenetic landscape has been rigorously formalized in the context of the systems dynamics state approach to the study of cell fate. Cell fate determination is predicted to exhibit certain dynamics, such as attractor convergence or oscillatory. The term epigenetic has also been used in developmental psychology to describe psychological development as the result of an ongoing, bidirectional interchange between heredity and the environment. Interactivist ideas of development have been discussed in various forms and under various names throughout the 19th and 20th centuries. An early version was proposed, among the founding statements in embryology, by Carl Ernst von Baer and popularized by Ernst Haeckel. A radical epigenetic view was developed by Paul Wintrebit. Another variation, probabilistic epigenesis, was presented by Gilbert Gottlieb in 2003. This view encompasses all of the possible developing factors on an organism and how they not only influence the organism and each other, but how the organism also influences its own development. The developmental psychologist Erik Erikson used the term epigenetic principle in his book Identity, Youth and Crisis, and used it to encompass the notion that we develop through an unfolding of our personality in predetermined stages, and that our environment and surrounding culture influence how we progress through these stages. This biological unfolding in relation to our socio-cultural settings is done in stages of psychosocial development, where progress through each stage is in part determined by our success, or lack of success, in all the previous stages. Contemporary Robin Holliday defined epigenetics as the study of the mechanisms of temporal and spatial control of gene activity during the development of complex organisms. Thus, epigenetic can be used to describe anything other than DNA sequence that influences the development of an organism. The more recent usage of the word in science has a stricter definition. It is, as defined by Arthur Riggs and colleagues, the study of mitotically or meiotically heritable changes in gene function that cannot be explained by changes in DNA sequence. The Greek prefix epi in epigenetics implies features that are on top of or in addition to genetics. Thus, epigenetic traits exist on top of or in addition to the traditional molecular basis for inheritance. The term epigenetics, however, has been used to describe processes which have not been demonstrated to be heritable, such as histone modification. There are therefore attempts to redefine it in broader terms that would avoid the constraints of requiring heritability. For example, Sir Adrian Bird defined epigenetics as the structural adaptation of chromosomal regions so as to register, signal or perpetuate altered activity states. This definition would be inclusive of transient modifications associated with DNA repair or cell cycle phases, as well as stable changes maintained across multiple cell generations, but exclude others such as templating of membrane architecture and prions unless they impinge on chromosome function. Such redefinitions, however, are not universally accepted and are still subject to dispute. The NIH Roadmap Epigenomics Project, ongoing as of 2013, uses the following definition. For purposes of this program, epigenetics refers to both heritable changes in gene activity and expression and also stable, long-term alterations in the transcriptional potential of a cell that are not necessarily heritable. In 2008, a consensus definition of the epigenetic trait, the similarity of the word to genetics, has generated many parallel usages. The epigenome is a parallel to the word genome, referring to the overall epigenetic state of a cell, and epigenomics refers to more global analyses of epigenetic changes across the entire genome. The phrase genetic code has also been adapted. The epigenetic code has been used to describe the set of epigenetic features that create different phenotypes in different cells. Taken to its extreme, the epigenetic code could represent the total state of the cell, with the position of each molecule accounted for in an epigenomic map, a diagrammatic representation of the gene expression, DNA methylation and histone modification status of a particular genomic region. More typically, the term is used in reference to systematic efforts to measure specific, relevant forms of epigenetic information such as the histone code or DNA methylation patterns. Molecular basis. Epigenetic changes modify the activation of certain genes, but not the genetic code sequence of DNA, the microstructure of DNA itself, or the associated chromatin proteins may be modified, causing activation or silencing. This mechanism enables differentiated cells in a multicellular organism to express only the genes that are necessary for their own activity. Epigenetic changes are preserved when cells divide. Most epigenetic changes only occur within the course of one individual organism's lifetime. However, if gene inactivation occurs in a sperm or egg cell that results in fertilization, then some epigenetic changes can be transferred to the next generation. This raises the question of whether or not epigenetic changes in an organism can alter the basic structure of its DNA, a form of Lamarckism. Specific epigenetic processes include paramutation, bookmarking, imprinting, gene silencing, X chromosome inactivation, position effect, reprogramming, transfection, maternal effects, the progress of carcinogenesis, many effects of teratogens, regulation of histone modifications and heterochromatin, and technical limitations affecting parthenogenesis and cloning. DNA damage can also cause epigenetic changes. DNA damages are very frequent, occurring on average about 60,000 times a day per cell of the human body. These damages are largely repaired, but at the site of a DNA repair, epigenetic changes can remain. In particular, a double-strand break in DNA can initiate unprogrammed epigenetic gene silencing both by causing DNA methylation as well as by promoting silencing types of histone modifications. In addition, the enzyme PARP1 ribose polymerase and its product polyribose accumulate at sites of DNA damage as a part of a repair process. This accumulation, in turn, directs recruitment and activation of the chromatin remodeling protein ALC1 that can cause nucleosome remodeling. Nucleosome remodeling has been found to cause, for instance, epigenetic silencing of DNA repair gene MLH1. DNA-damaging chemicals, such as benzene, hydroquinone, styrene, carbon tetrachloride and trichloroethylene, cause considerable hypermethylation of DNA, some through the activation of oxidative stress pathways. Foods are known to alter the epigenetics of rats on different diets. Some food components epigenetically increase the levels of DNA repair enzymes such as MGMT and MLH1 and P53. Other food components can reduce DNA damage, such as soy isoflavones and bilberry anthocyanins. Epigenetic research uses a wide range of molecular biologic techniques to further our understanding of epigenetic phenomena, including chromatin immunoprecipitation, fluorescent in situ hybridization, methylation, sensitive restriction enzymes, DNA adenine methyltransferase identification and bisulfite sequencing. Furthermore, the use of bioinformatic methods is playing an increasing role. Computer simulations and molecular dynamics approaches reveal the atomistic motions associated with the molecular recognition of the histone tail through an allosteric mechanism.